For regeneration to occur, cells need to proliferate, and for cells to proliferate, they need to grow. And interestingly, ribosome biogenesis actually antagonizes cell cycle progression until the cell has grown to an adequate size. And the first step in making ribosomes is to transcribe ribosomal genes, or rDNA genes. So what are the differences in ribosome biogenesis between regenerative and non-regenerative organisms? So humans have about 600 copies of rDNA genes per diploid cell, and these are organized as tandem arrays, so one gene immediately after the other. Its repeat is 40 kb in length, and it contains the coding sequences for 18S, 5.8S, and 28S, followed by a large intergenic spacer of about 30 kb in length. And this is where most of the regulatory sequences are located, such as promoters, enhancer elements, and terminator elements. And UBF is a nucleolar transcription factor that binds ubiquitously to active rDNA repeats and recruits POL1 transcription machinery to the promoter site. UBF is a very large protein of about 764 amino acids in length. It has a dimerization domain, an acidic C-tail, and 6 HMG boxy domains that are used to bind DNA. And this is the canonical architecture in humans, frogs, birds, and all other higher eukaryotes. So currently, we are finishing the genome and transcriptome assembly of Hydrotinia, which is a highly regenerative marine cnidarian. And by assessing its scaffolds, we identified the rDNA genes are also organized as tandem arrays as in the human genome, but the size of each repeating unit is much, much smaller. It's about 7 kb. And while the size of the rDNA components are roughly the same size as the human ones, it's the size of the IGS that is much, much smaller in Hydrotinia. It's less than 400 base pairs compared to 30 kb in humans. So we hypothesized that the control elements that each human repeat carries are actually in the junction sequence before the first repeat in the rDNA arrays in Hydrotinia, and so the entire cluster gets transcribed in one go, enabling Hydrotinia to cope with a high demand for ribosomes during regeneration. And Hydrotinia also does not seem to have the canonical UBF protein, so we think this might be a novel mechanism, a novel protein as a nucleotranscription factor in Hydrotinia. So currently, we are in the process of identifying this protein and assessing its cellular function and its contribution to regeneration in Hydrotinia. And we also want to identify where in the tree of life UBF first emerged